Hello, this is lecture number 35 in the series of talks on biochemistry. These are not from Lehninger per se, but they are discussions of various investigators who helped create the discipline of biochemistry that we know today, and we are taking these in chronological order. Uh, Many of these have won Nobel Prizes when the Nobel Prizes started a bit over a century ago. The earliest uh, uh, personalities who were involved in this obviously were not able to apply for that, but in many cases, in the latter group, these are Nobel Prize laureates in medicine, physiology, and one famous biochemist additionally has a Nobel Peace Prize, which would be his second. So we begin all the way back to 1747. James Lind was the first person to connect the incidence of scurvy with seagoing sailors to vitamin C deficiency, which led to the necessity to bring fruit and dried fruits on board. Antoine Lavoisier discovered oxygen and energy transformation into heat. Louis Pasteur began with crystalline studies on tartaric acid, and then later on discovered ferments in alcohol production. J. Willard Gibbs was the first in 1873 to have the discovery of free energy and is the father of thermodynamics. Johann Thudekim discovered sphingolipids. Edward Buchner, yeast fermentation rather. Arthur Hardin and William Young were early investigators for glycolysis and phosphorylated intermediates. We then go to Archibald Hill, who developed hemoglobin cooperativity for oxygen. Leonore Michaelis and Maud Menton for enzyme kinetics. Frederick Banting and Charles Best discovered insulin treatment for diabetes. James Sumner discovered that enzymes are in fact proteins. Otto Warburg, tumors increase glycolysis. Hans Krebs, the urea cycle. He'll come in later on in this discussion. Gustav Emden and Otto Meyerhoff for muscle fermentation. Frank Strong, D. Wayne Wally, and Conrad Elvehjem discovered niacin and role in dehydrogenases. Hans Krebs also discovered a tricarboxylic cycle, which is named after him. George Beadle and Edward Tatum defined the gene. And then vitamin K was discovered by Hendrik Dahm and Edward Doisy. Dorothy Hodgkin discovered the structures of penicillin and cholesterol. Carl and Gertie Corey discovered glycolysis. Barbara McClintock, genetic recombination. Albert Lehninger discovered mitochondria or the site of beta oxidation. Dorothy Hodgkin additionally discovered the structure of vitamin B12, which is a very formidable molecule. It was synthesized by Robert Woodward of Harvard University and was a singular feat of organic chemistry and arguably one of the greatest organic chemists in history. Melvin Calvin discovered carbon fixation in plants. I had the honor of meeting him at a lecture at university. Maurice Wilkins and Rosalind Franklin performed X-ray DNA X-ray crystallography and were very important for the development of that of the structure of DNA. Linus Pauling and Robert Craig developed the alpha helix and beta sheet structures of protein. Erwin Chargaff, who had Chargaff's rules for DNA stoichiometry. The number of adenines is equivalent to thymines, etc., etc. Frederick Sanger developed amino acid sequencing. And then the double helix of DNA was James Watson, Francis Crick. One thing I will mention about the Nobel Prize, it was awarded for the DNA structure to Drs. Watson, Crick, and his X-ray crystallographer, Maurice Wilkins, was that this was one of three teams that were in a fierce, intense race to determine the structure of DNA. You not only had to have the right data, you had to have the right interpretation. So the other two groups that were involved, one was headed by Rosalind Franklin, who had the correct data, but not the correct interpretation. And the other group was headed by Linus Pauling, who had neither the correct data nor the correct interpretation. Watson and Crick and Wilkins had both of these. They were first to the finish line, and they had the Nobel Prize. There was, of course, controversy about Rosalind Franklin. One thing that is notable is that Rosalind Franklin and Maurice Wilkins were not friendly colleagues. There was some hostility between them. And even so, Nobel Prizes go to a maximum of 3 investigators for any given project, and the Nobel Prizes go to investigators who are living. And if memory serves, Watson, Crick, and Wilkins got the became Nobel laureates in 1962, and Rosalind Franklin had passed away of cancer in 1958. I recognize that this is a controversial point, but this is a view that I think is defensible on those levels. John Buchanan, 1953, to synthesize nucleotides. Arthur Kornberg did likewise for DNA. He discovered DNA synthetase. Earl Sutherland discovered cyclic AMP, the second messenger. Hans Krebs again discovered the glyoxylate cycle. Jens Skou discovered sodium-potassium ATPase. Max Perus and John Kendrew discovered hemoglobin. Francis Jacob and Jacques Monod discovered bacterial gene expression. Robert Hawley developed the first transfer RNA sequence. John Vane, non-steroidal inflammatory drugs were developed by him. Peter Mitchell developed the chemiosmotic theory of energy production. In 1966, Hargobind Khorana of University of Wisconsin developed a genetic code. One of his postdocs was my major professor under Dr. Khorana. And while I'm at it, I can say that the other Nobel laureate I met was James Watson. He was the head of the Cold Spring Harbor Laboratory. There were symposia that were present throughout the year there, and all the young investigators, scientists and would-be scientists met James Watson during the introduction plenary. So that was very memorable, obviously. I remember it all vividly, quite vividly, and Dr. Calvin also. Masayasu Nomura discovered ribosomal subunits. David Baltimore and Howard Temin reverse transcriptase. Baltimore, I believe, was the youngest of the Nobel laureates at the time. He was in his early 30s, early to mid-30s. George Kohler and Cesar Milstein discovered monoclonal antibodies. Soon Bergstrom and Brent Samuelson developed prostaglandins. Akira Endo developed statins, which are used to reduce cholesterol. They're different classes and varieties. Thomas Keck was RNA introns and exons. Jean-Pierre Sauvage, Ben Farigna, and Fusen Studdard were macromolecular applications to nanotechnology. In 1984, Carol Greider and Elizabeth Blackburn discovered telomerases. Francis Arnold, George Smith, and Gregory Winter, bacteriophage and enzyme production. In 1986, Thomas Liddell, Paul Modric, and Aziz Sankar discovered DNA repair. Thomas Sudhoff, Randy Scheckman, and James Rothman, membrane transport. Craig Mello and Andrew Fire discovered RNA interference in gene splicing. James Thompson for stem cells. Francis Collins and J. Craig Venter for the human genome. And the most recent discovery of note, Jennifer Doudna and Emmanuel Charpentier for in vitro gene modification by CRISPR technology. These are some of the big names, and they're just incredibly impressive people with the incredibly elegant experimentation. And in my view, these individuals have improved the state of humanity and increased our understanding and knowledge. Admittingly, there was some narrative. I did focus on the mechanistic qualities of the pathways. It would be a talk that would extend into low 3 digits had everything that was conducted by pros been placed in diagrammatical form, and there was no need nor desire to overload anybody with the say the pathway of lysine production. These talks are meant to be springboards, and they can be looked at. I will say that if you can find it, the most elegant biomechanical reaction mechanism is thiamine pyrophosphate in the production of pyruvate dehydrogenase in the first of the 3 enzymes in the triplicate formation of the production of acetyl-CoA from pyruvate. That's an incredibly elegant and extremely difficult reaction mechanism to suss out, and that would be my own choice as the most intriguing reaction in this. The rest of the material is springboard to further study and at least acquire a vocabulary. So I appreciate giving this series, and hope it's of some value to anyone who would take it in and give it a fair hearing. Thank you very much. That's it.